Welcome back. So I just appeared because I look hideous today, and you guys should have already seen how to make your foldable. So at this point, go ahead and copy down the learning goals. This is a new week, week 5, so this should be a new sheet of paper. Copy down these learning goals. Feel free to pause while you do that, but be aware that these are the learning goals for the day. So welcome to the cell cycle. Okay, we're going to be talking about basically how we can get more cells. Now, if you'll recall, we have 3 parts to the cell theory. Number 1, all living things are made of cells. Number 2, cells are the basic unit of structure and function. And number 3, all cells come from other cells. And that's the part we're going to focus on today. Now, if all cells come from other cells, what does that mean? They can't just pop right out of thin air. They're not popcorn. Okay, they have to come from somewhere. Okay? Now, it's also important to note the difference between prokaryotes and eukaryotes. Now, prokaryotes are simple cells. They're like our bacteria. They're unicellular, and so they can just simply split in 2 in order to copy themselves or to make more. Eukaryotes, on the other hand, are complex, and they have to be able to copy their DNA and all their organelles, especially if they're specialized, right? Can't just use a brain cell for a muscle cell, okay? So we have to be able to have a process of making this. So here's a little bit on DNA. DNA we're going to cover more in the genetics and DNA units, which are to come. But here's a little brief lowdown that you'll need in order to understand the cell cycle. Okay? Now, DNA is stored in the form of chromosomes. Okay? They're basically like bundles of DNA that look like the shape of an X. Okay? So humans have 46 chromosomes. Seahorses have 48, and the great white sharks is 82. They're unique to each species, which would make sense why, if you're not the same species, you can't reproduce. You guys don't have the same amount of DNA, so then you would just have this whole big mix-up of things. Chromosomes are only visible during cell division. Before that, they're in this like spaghetti-like form called chromatin, and they're not visible to the naked eye, so you can't see them under a microscope. And this chromosome structure is unique because it allows us to divide our cells without losing any of our DNA. And during cell division, each of these chromosomes will consist of 2 sister chromatids, and I'll show you what that will look like next. So here you have our chromatid. This is what your DNA would look like if it's not about to divide or copy itself, okay? Now, in order to make a new cell, you have to copy the DNA. You can't just divide your DNA in half, or else eventually we'd run out of DNA. So it makes a copy of itself, and then it looks like this. We have 2 sister chromatids, so one here and one here. All right, and so when it condenses to be a chromosome, we actually can see it because it's actually thicker, right? Condensed meaning smaller and more thick. So these are the events of the cell cycle. What I want you to do is draw this on the back of your foldable. Okay, draw this circle and these proportions of the circle. It's important because these represent how often your cells are in each stage. Okay? Biggest one is interphase. We're going to go over this in a moment, along with mitosis, and then finally cytokinesis. And the end result of the cell cycle is 2 identical cells, right? It's important to remember, mitosis creates 2 identical cells. So now as you go and take notes on mitosis, this is what where everything's going to go. So on the front flap of your foldable will be anything here. These are going to be titles and a couple pictures. So on the front flap is going to be here. The inside of your foldable, the information where you open the door or open the flap, is going to be on the right-hand side. Okay? So if you have any questions, I am in the room, so feel free to come up and ask. But make sure you're doing this correctly. So the first stage of the cell cycle is called interphase. So I want you to write the big I on the top. Below would be interphase, and I want you to draw this picture. It's a really good one. And in interphase, you're not technically in mitosis yet. This is like the cell just doing its jam, all right? And inter, in case you didn't know, meant in between. So it's like the in-between phase. You're not dividing yet. You're in between divisions, okay? And cells are going to be in this phase for most of their lifetime. Cells aren't just constantly dividing, or else we would constantly be growing, or organisms would be constantly growing. Okay, so during interphase, you're going to see a couple of things happen. Number one, the cell's going to grow. Okay, this is known as G1, or growth number 1. Okay. Number 2, the DNA gets copied, which is called the S phase. Not really important why it's an S, but it's an S phase. DNA gets copied because you can't just divide your DNA along with the cell. You have to be able to divide it. And finally, centrioles, these get copied in something called G2, so the second growth phase. And so in essence, what interphase is just the cell preparing to divide. All right? So in order to prepare to divide, to have two identical cells, you have to grow and increase the amount of stuff you have. You have to copy your DNA, and you have to copy these things called centrioles. So if you're looking in the picture, these are them, and they're basically these organelles that help in the mechanism of dividing. And so you'll see those coming up a lot more. The first actual step of mitosis, so wink wink, nudge nudge, be able to know that interphase is the first stage of the cell cycle, but prophase is the first stage of mitosis. That might be worth noting. Okay? Prophase is the first step of mitosis. So in this, I want you to think as pro as in prep, or before. Okay? Remember, prokaryote would be before or early cells. Prophase would be early stage of mitosis. In mitosis, our DNA is going to condense to form our chromosomes. So if you take a look at the picture on the right, we see our X-shaped DNA chromosomes. Second thing you're going to see is that centrioles are going to move to the opposite sides of the cell. So those little bead-like divider organelles, basically, you'll notice are here and here. They're going to move to opposite sides of the cell. I want you to think, why would they want to be on the opposite sides of the cell if they're going to help divide? Well, that's because they're going to pull the cell apart. They're going to pull it apart into two separate cells. Okay, so they need to be on opposite sides to basically pull it in half. Next, you're going to have something called spindle fibers form a bridge. If you're looking in this picture, the spindle fibers are the long lines here. Okay, and they also are going to help. Think about if you were in a tug-of-war, you had a rope in between, or if you're trying to help pull something out with a large rope, that's what these are. So the centrioles are going to use these spindle fibers or these ropes to help pull the cell in half or divide it in two. And finally, the nucleus breaks down. Now it's important for the nucleus to break down so that we can actually divide the DNA. Remember, the nucleus's function is to protect the DNA at all costs. It's going to protect it so that no pathogens get in and nothing can use it in the wrong way. But if we're going to divide the DNA with our cells, we have to get rid of that nucleus first. So it's going to break down. The third stage of mitosis is metaphase, okay? Think like meta world peace, okay? Funny joke, I like the Lakers, remember, so don't think too much into that. But meta actually means middle. So meta Earth would be middle Earth. If you ever see this prefix, it's referring to middle or half. So this would be referring to the chromosomes lining up in the middle. Notice how there's no nucleus now. We see our centrioles and still on the opposite side of the cell. Spindle fibers are attaching to our chromosomes and they're lining up in the middle. This is going to make it so that these can go to opposite sides and divide in two. We're physically pulling the cell apart in opposite directions. Okay? Now, metaphase plate in this picture here is referring to chromosomes lining up in the middle. This region here is called the metaphase plate. Okay? So again, meta means middle, and in this phase, the chromosomes are going to line up in the middle, with centrioles on either side, and spindle fibers attaching to something called the centromere, which is just the middle of these chromosomes, the middle of the X. Next, we have anaphase. I want you to think of anaphase or remember it by A like away. So anaphase, the away phase. And here, this is where the chromosomes split at the centromere and move to opposite sides. So take a look at the picture here. The centromeres, the centrioles rather, are still on opposite sides. Spindle fibers are still attached at the centromere, but now they're physically pulling them apart and ripping them in half as they move to opposite sides. So this is the beginning of the actual division part. Notice how it's moving away, and we can see the beginnings of new cells form. Telophase is step 4 of mitosis, and I want you to think of telophase like teleport, or to move. And this is where the chromosomes spread out within their new regions, and we start to see the nuclei form again. So if you look at this picture, the 2 new cells are basically formed but not separated yet. Our centrioles are now just chilling in the cells. Spindle fibers have disappeared. We have our chromosomes spreading out. They're not so clumped together. And we have the nuclei starting to form. So that's what this gray circle looks like here. Okay. Now the last step we're going to talk about is not part of mitosis either, right? Light interphase. It's not part of mitosis, but it's part of the cell cycle, and that's cytokinesis. Cytokinesis. It's basically a big fancy word for when we're going to split these cells apart. Okay, so the cell membrane is going to pinch at the center, and each cell is going to have now the same number of chromosomes. So here we were at telophase, okay, with a little bit of attachment in between. Our nucleus reformed, and then with cytokinesis, we basically cut the cord, and now they're separate. All right, so now we're at 2 separate cells, completely identical, same number of chromosomes, do the same function. There's no difference here. Now, I know that's a lot of information, but I have a great analogy that goes with it. So we're going to scroll back up to the beginning. So interphase, this is where the cell spends most of its time, all right? It's just chilling. This would be like if you were in a relationship with a boyfriend or a girlfriend. The majority of the time, you're not breaking up. Most of the time, you're in a relationship or single. And right now, they're chilling. We are growing as a couple. There's 2 of everything, right? His and hers towels, his and hers whatever, all right? And now, prophase is like the initial breakup. You're breaking up the nucleus, you had the fight, things are in turmoil, and now your centrioles and your spindle fibers are like your friends, and they're like, oh no, we gotta pull these 2 apart. He's not good for her, she's not good for him, so we're gonna break these people up. Metaphase is when you guys come together and you get your stuff back, right? His sweatshirt goes back to him, and her keychain goes back to her, or Tupperware you guys shared food in, or whatever. But you're dividing up your stuff, and your friends are your centrioles and your spindle fibers here, really ready to help pull you guys apart, make you single again. They don't think you guys are good for each other. Now, anaphase is when your friends finally pull you away from one another. You guys have your stuff back. There's no turning back. You guys are now officially broken up. And it's time to be your own people again with your own friends, okay? Telophase is like when you're back to singledom. It's new, you're not used to it, maybe you're thinking about your ex, but you have your friends with you, and they're going to help you get through this. And cytokinesis is the stage where you're completely single. No more remembrance of them, you're moved on. I hope that analogy helps with my former students. If you want to re-listen to this, I recommend you kind of go back and through, fill in anything, but also maybe add the analogy of the breakup. It kind of makes sense. So maybe fill in your notes with that. We do have one more video, so click on part 3.